My name is Dr. Jonathan Miner. I'm an associate professor of medicine in the Division of Rheumatology at the University of Pennsylvania Perelman School of Medicine. I also direct the RVCL Research Center at Penn. My goal in this video is to provide an update on our development of therapies for retinal vasculopathy with cerebral leukoencephalopathy, RVCL. And in particular, in this video, I want to walk you through a few different aspects of how we do our work. First, I want to tell you a little bit about the support that we have received, and not just from within the institution, but also from outside the institution. And then I want to transition to explain how that support is translating to real productivity that we believe will lead to effective therapies to slow down this devastating disease and even a cure. So first, I want to tell you a little bit about the Penn Colton Center for Autoimmunity. So many of you probably know I started my career at another institution, and I ended up moving to Penn about 2.5 years ago. This move was absolutely revolutionary for our ability to develop therapies for RVCL. And here I want to highlight one aspect of how that move translated to additional support for developing therapies. So on the left here in this photo are pictured Judith and Stuart Colton. They donated $10 million to Yale, $10 million to Tel Aviv University, $10 million to New York University, and $10 million to Penn in order to start an initiative for autoimmune disease research. Then they donated another $50 million to Penn, so $60 million in total, to further advance autoimmune disease research. And the concept here is that there has been a war on cancer. Why can't there be a war on autoimmunity? Their donation has led to a revolutionary emphasis on autoimmune disease research at multiple institutions who work in collaboration as part of the Colton Center Consortium. You can see on the right here, we had the Penn Colton Center Symposium where I spoke. Their donation, in part, 150,000 of it, went directly to developing therapies for patients with RVCL. I spoke at another consortium event in New Jersey last year, and after my talk, Stuart came up to me, Stuart Colton, and asked me more about the disease, what it's like, how it affects families. And I told him how grateful people are for their support for our work in developing treatments for RVCL. And it was a very touching moment for me, but also I think for the Coltons. So this is one of many examples of how this move has led to direct support for developing therapies for this disease. And I'm going to tell you more about that. In addition, so the laboratory right now is in a building just behind what they call the quad here. We're in one of these buildings back here. We are going to be moving to a new laboratory research building that is currently under construction next year. And there are a couple of floors there that are designated for autoimmune disease research, and we're going to be at the center of this Colton Center for Autoimmunity in this new building, which is exciting for the whole lab to be in a new space. And I think it's also a reflection of the support the institution is providing to our vision and our work. As another example, so Penn has an interim president, Larry Jameson, who's a physician scientist. He was the dean of the medical school for many years. And for perspective, he is very well aware of this research program. He has met with me about it and with representatives of the Clayco Foundation, and very directly expressed support for the work we are doing. So I think there's widespread awareness at the institution about the importance of this disease and the urgency with which we must develop a therapy. In addition to that, Drew Weissman and Katie Carrico, who've both been affiliated with Penn in the past, Drew is still affiliated at Penn, won the Nobel Prize last year for their work in modified RNA, which became important in the context of vaccines, but is also important in the context of gene therapy. Now, this office was Drew's office when he made that initial discovery and reported it of modified RNA. And Drew is now helping us with our gene therapies for RVCL. Been overwhelmingly supportive of the work we are doing, and we are very grateful for that. But I think it gives you, I'm starting to try to paint a picture of the broad impact, the people involved in this work, and the list goes on and on and on. I really don't have time to list everyone who's helping. It's been amazing. I'm speaking at the Yale Innovation Summit in May, and in particular going to highlight the medications, the drugs that we're developing for RVCL, which incidentally are likely to have applications beyond RVCL, including in diseases like cancer, infectious diseases, other autoimmune diseases. And so again, I want to highlight that while we're trying to cure RVCL, a key concept here is that the work we are doing is likely to benefit people with common diseases as well. So as I've often mentioned in my videos, there are 3 billion bases in the human genome, A's, T's, C's, and G's. And RVCL can happen because of just one extra letter in the genome within the TREX1 gene. We have developed gene therapies for this. The improvement in these gene therapies has been exponential. And I'm just going to highlight this. I realize that looking at data like this may be confusing, but I'm just going to try to walk everyone through this to make sure that it's well understood. So in these cells, we have the full-length TREX1 protein. And in these cells, we have a truncated mutant TREX1 protein that causes RVCL. And in the right, we are detecting the presence of the normal protein. And what you can see is with our first generation gene editor, we were able to increase the percent of normal cells that have normal TREX1. So we're talking about fully correcting the mutation from 6% to 10%. In our second generation gene editor, we're able to increase it from 6% to 42%. Now the full length is 63%. So we're almost 2/3 of the way there. So now you can start to imagine, this is just within the last year, we've had this exponential improvement. So of these, so basically if we're able to deliver the editor into a cell, 2/3 of the cells end up correcting back to the full-length normal version of TREX1. This is incredibly exciting, and we think it's going to get better and better and better. And we're also working on delivering this in vivo, which means in animals. So there's a couple different ways that can be done. One way that can be done is using a virus. So viruses are very, very efficient at delivering DNA into a cell. So what we do is we modify a virus called AAV such that it includes this gene therapy. And this is a very efficient way of delivering gene therapies to the retina. As an example, the first FDA-approved gene therapy was developed here at Penn, and it used an AAV-based system. And we work with the people who developed that initial gene therapy. So we're working with people who've done this before. In addition, gene therapies can be delivered in nanoparticles that contain RNA, in this case, RNA encoding a gene editor, which can correct the abnormal DNA sequence that causes RVCL to the correct DNA sequence, and therefore the abnormal protein encoded by the DNA to the correct protein. And if we do that, the toxic protein will be gone, and there's a chance that we may be able to cure the disease if we can deliver this to the correct cells in the body. Now, I do not want to convey the idea that this is easy, but I was heartbroken not too long ago when a patient reached out to me and said that they heard that we had failed at developing our therapies. And that compelled me to make this new video to communicate to the world that that is not true. It's quite the contrary. Things are moving forward exponentially, and I have great hope that we're going to be able to find effective treatments. I don't want to overpromise because I can't guarantee when this is going to happen, but someone had said to me that they thought it would be 20 years before it happened, and I don't think it's going to be 20 years. I'm thinking on a much shorter time scale than that. But how fast this goes is not entirely predictable. What I can guarantee is that we are doing everything possible to make sure it happens as fast as possible. So the other thing that's going incredibly well is our development of small molecule drugs. So you can small molecule drugs are chemical type drugs that are synthesized by chemists. And anytime you take a pill, that's a chemical type drug, a small molecule drug. So these are synthesized by chemists, and this is not easy either. In many cases, we have to screen millions or even billions of compounds to find the best ones. And in order to do this, they have to go through a pipeline, which is illustrated here, until we reach the point where we have an effective drug, which can be taken as a pill. So how do small molecule drugs work? So you take it as a pill, the small molecule is absorbed, and they will actually stick to the protein and prevent the protein from doing damage. That's the general concept. And so this sounds like a simple idea or a simple concept, but it's not simple, and I'll try to illustrate that. So initially we have to screen, and there's different ways that people screen. People have screened using computers, artificial intelligence, or actual screens where we measure the ability of the TREX1 protein, the abnormal TREX1 protein, to pull out drugs. And so you can kind of imagine a tube that has literally billions of chemicals in it, and we pull out the chemicals that stick tightly to the TREX1 protein. And then how do you figure out which chemicals are stuck to it and so on? It's a very complex process, but we have ways of doing this, and we have done this. This has been done. Then come the next steps. Chemists have to synthesize those compounds in larger quantities such that they can be studied. Again, not a simple process. It's basically, this is about brute force. And then we have to screen them. So our collaborators screen them. We screen them in our lab as well. So screening them is also a rather complex process. We screen in different ways. We screen the ability of the drug to interact with TREX1 directly, their stability measurements. We also determine whether the drug blocks the toxic activity of the RVCL mutant TREX1 protein. So blocks the toxicity caused by the abnormal protein in cells that express that have the human gene and the human protein. So there's a variety of screening approaches that have to take place. And then we have to figure out which drugs block and which drugs might have some toxicity. Again, it's a huge amount of work, but we are making progress. We have identified compounds that block the toxicity of the abnormal TREX1. They exist already. We are working on making better ones. I'll explain some of the limitations, but again, I want to highlight this is just the kind of a cursory summary of everything that needs to be done. There's far more than this, but I feel like I need to convey how complex this is, but I also want people to know that we are having success. So another component is crystallography. So in crystallography, you take your protein and you can soak it in different chemicals, so drugs that bind to TREX1, shoot X-rays through it, and the X-rays will diffract, and a computer can take a diffraction pattern of the X-rays shooting through your protein crystals. They can actually make a picture of TREX1. Now, this is not TREX1, but it's a protein, and it's meant to illustrate the concept that we can actually take pictures of these proteins, even though they're incredibly tiny, by forming crystals, shooting X-rays through them, and pictures of the drug bound to the protein, which is super important because it allows us to design better drugs when we see how our initial candidate drugs interact with TREX1. This is something we've already done, and we are using this. And then we have to go back and the chemists synthesize more compounds, and we screen again to find which ones work best. So it's an iterative process as we make things better and better. So we are moving as fast as we can, but we also need to make sure we do this safely and responsibly. I want to say thank you to everyone who's supporting our work. I strongly believe that, so a lot of people think that scientists are just, smart people working in a lab. My view is that a lot of this stuff is just a function of elbow grease, hard work. We know the things that we need to do. We need people to do the work. And so I want to communicate about the things that help us go faster. So first, patient and family support makes a huge difference. We've had lots of patients and families come through the lab, and that has been amazing because it's very motivating to everyone in the lab. I posted another video just a moment ago where I talked about how we have a board with all the patient, the RVCL family mutations. And I said, when I look at these, I see people's faces, the patients and family members' faces. And members of the lab might see them as letters and numbers on the board, where they look at, the DNA sequence or the protein sequence on the board. But when patients and families come by the lab, it makes a huge difference because the lab members recognize that these aren't just letters and numbers. These are real people who have a currently untreatable disease, although we're going to change that. So the other thing is we've had great donor support, and we're very grateful for that. We're grateful to Clayco Foundation, the yearly event at Illumination. And there have been a lot of families that have chosen to support the work we're doing. Like I said earlier, I moved here because I wanted to develop a treatment. That's why I moved to Penn, and the institution has supported us in doing that. And we are actually doing it now. So it's very exciting. So what helps us go even faster? Like I said, it's a function of how many people can do the testing and how many people are working at the bench in the lab. So in my other video, I showed you the benches and where people are working. More people we have at the bench actually doing the work, the faster it's going to go. We are working toward a cure. You can reach out to us anytime. There are websites and phone numbers here if you need to reach us. I also want to highlight that we have our RVCL coordinator who joined us recently, Ashley Batista. So Ashley actually worked with me for many years in St. Louis, called me up late last year and asked to work with us again. It's been amazing. She works super hard. She's very accessible, and she's here to help. Please feel free to reach out to me anytime. And most importantly, I want to thank everyone who's supporting our efforts. we are going to develop a treatment for this disease. It's going to happen. And it's just a matter of how quickly we can do it. It's not going to take us 20 years, but I want it to happen much, much, much faster than that. I'm very concerned about patients living with this disease right now and finding a way to stop it before it's too late for them. There's also future generations who are affected by this disease. We are finding more and more patients and families every single month. Just for perspective, the number of families and patients that we know of has more than tripled since I moved here a little over 2 years ago. And I think part of that is people reaching out to us. Another aspect of it, though, is that genetic testing is becoming more common, not just in the United States, all around the world. And so thank you to everyone who is supporting our efforts. And our commitment to you is that we're going to work day and night till this problem is solved. We know the genetic cause of this disease. We understand now the toxicity caused by the abnormal TREX1 protein. That's been another big revolution in developing a treatment. We can read out that toxicity and we can block it with some of these drugs. Now it's a matter of finding drugs that stay in the body long enough, that are non-toxic, that are safe, and finding a way to deliver the gene therapy efficiently enough such that we can cure the disease. So we know we can correct the mutation in cells in addition in the lab, and we are working as we speak. We have mice with the human gene in them that we made here at Penn. And we have been testing the gene therapy in mice to find ways to make it more and more efficient such that we can correct the mutation in different cell types in the animals. That is what is underway currently. That is a direct result of small and large donations that are facilitating our work. So thank you to everyone. My commitment to you is transparency, that every dollar that is donated to this cause goes to the work that I've described. Thank you to everyone who's been supporting us in this effort.